Social dominance, CSIRnet Life Sciences Lecture. Social dominance refers to the hierarchical organization of individuals within a group based on access to resources such as food, mates, and space. These dominance relationships are usually established and maintained through direct interactions like aggressive encounters, displays, threats, and submissive behaviors. Over time, stable hierarchies emerge in many social animals, reducing the frequency of actual conflicts and establishing predictable patterns of behavior. Dominance is not just about aggression, it reflects a socially recognized rank that is reinforced through repeated interactions and social signals. Dominance hierarchies can be linear or nonlinear, stable or dynamic, and may differ between sexes, age classes, or seasonal contexts. In a linear hierarchy, also called a pecking order, each individual dominates those ranked below and submits to those ranked above. For example, in chickens and wolves, linear dominance orders are common, leading to efficient resource allocation without constant fighting. In nonlinear hierarchies, dominance relationships are more complex and may involve triangular or looped interactions, where A dominates B dominates C, but C may dominate. These are more common in species with complex social systems or changing group compositions. Dominance can be context dependent, where an individual may be dominant in one context, e.g., feeding, but subordinate in another, e.g., mating. It may also be absolute or relative. A dominant individual in one group may not be dominant in another. The factors influencing dominance include body size, strength, age, experience, prior victories, winner effect, hormonal state, especially testosterone levels, and even social alliances. In primates, for example, individuals often gain and maintain high rank through coalitions and alliances, not just brute strength. The advantages of dominance are significant. Dominant individuals typically enjoy greater access to food, preferred mating partners, safer resting sites, and higher reproductive success. In many species, dominant males father more offspring, while dominant females may have priority access to food, enhancing their reproductive output. However, dominance also comes with costs, such as higher energy expenditure in maintaining status, greater risk of injury during challenges, and elevated stress levels, particularly in unstable hierarchies. In contrast, subordinate individuals may adopt alternative strategies to survive and reproduce. These include sneaky mating tactics, delayed reproduction, or emigration. Some species exhibit conditional strategies, where individuals switch roles based on environmental or social cues. For example, in some fish species, smaller males mimic females to sneak fertilizations without engaging in direct competition with dominance. Dominance hierarchies may be maintained through ritualized displays and signals that avoid costly fights. These include threat gestures, postural displays, facial expressions, and vocalizations. For example, baboons display threatening yawns showing canines, while wolves use tail posture and eye contact to assert rank. Submission signals, like grooming, crouching, or avoiding eye contact, serve to reinforce the social order without escalating conflict. In primate societies, social dominance is often closely tied to coalitionary behavior. Males and females form alliances to defend or contest status. High-ranking individuals often use social bonds, grooming networks, and political strategies to maintain rank. For instance, in chimpanzees, males that form long-term alliances are more successful in attaining and holding dominant positions. Dominance also influences stress physiology. Subordinates in unstable hierarchies often have elevated glucocorticoid levels, which can impair health and reproduction. Some species exhibit sex-specific hierarchies, where males and females maintain separate ranks. In matrilineal systems, such as in spotted hyenas and some primates, dominance is inherited maternally, and daughters often assume ranks adjacent to or just below their mothers. This inheritance allows for social stability and predictability of rank across generations. In species like elephants and meerkats, matriarchs or dominant females exert strong control over group decisions and reproduction. Dominance can also have evolutionary implications. Traits that enhance dominance, such as large body size, ornamentation, or aggressive behavior, may be favored by sexual selection. In many cases, dominance rank correlates with fitness, especially in species where dominant individuals monopolize reproductive opportunities. However, selection also maintains diversity in social strategies. Subordinates may contribute genetically through alternative tactics or through kin selection by supporting relatives. In social insects like bees and ants, dominance is not always expressed through physical aggression but via chemical signaling and reproductive suppression. In queen-dominated colonies, pheromones suppress worker reproduction, enforcing dominance through physiology rather than behavior. In humans, dominance is expressed more subtly, often through verbal cues, posture, eye contact, and social influence. While physical aggression is less common, psychological dominance and leadership roles can influence access to resources, decision-making power, and reproductive success. Human dominance hierarchies are more flexible and culturally influenced but still share core features with animal models. In summary, social dominance is a fundamental organizing principle of animal societies, affecting access to resources, stress levels, social bonds, reproductive output, and survival. Its expression varies across taxa, but its adaptive value is universally significant. A clear understanding of dominance patterns, mechanisms of maintenance, and evolutionary consequences is essential for cracking questions in CSIRnet Life Sciences. CSIRnet Previous Year Questions. Social Dominance. CSIRnet Deck 2018, Part C. In a group of primates, researchers observed that individual A consistently displaced individual B from food sources, but avoided individual C. Which of the following is the most likely explanation? AA is subordinate to B and C. BA is dominant over B but subordinate to C. CB is dominant over A but subordinate to C. DC is dominant over both and B. Answer, B. Explanation. Displacement indicates dominance, avoidance indicates subordination. CSIRnet June 2016, Part B. In spotted hyenas, females are dominant over males. Which of the following best describes this type of social system? A male-biased hierarchy. B. Egalitarian system. C. Matrilineal dominance. D. Random dominance order. Answer, C. Explanation. Female dominance passed through maternal lines defines a matrilineal hierarchy. CSIRnet Deck 2020, Part C. Which of the following is most likely to reduce the cost of maintaining dominance in a group living species? A. Frequent physical fights to assert dominance. B. Mation of stable linear hierarchy. C. Constant monitoring of subordinates. D. Isolation of dominant individuals from the group. Answer, B. Explanation. Stable linear hierarchies reduce physical conflict by establishing predictable relationships. Let me know when you're ready for the next topic. Shall we proceed to territoriality, or mating systems and reproductive strategies? Next. You said. Use of space and territoriality. ChatGPT said. Here is your CSIRnet JRF Life Sciences Ultra Detailed Lecture on use of space and territoriality, written at the JRF level, in paragraph-only format, maximum word limit, and without tables. This lecture includes advanced behavioral ecology concepts, tailored to CSIRnet, and features 3 previous year questions with answers in years mentioned. Use of space and territoriality, CSIRnet Life Sciences Lecture. In behavioral ecology, space use refers to the way animals organize, partition, and defend areas within their habitat to fulfill essential needs such as feeding, mating, resting, and raising offspring. One of the most ecologically and evolutionarily significant strategies animals use to regulate access to space is territoriality. Territoriality is the active defense and maintenance of an area, a territory, against conspecifics or sometimes heterospecifics, to secure exclusive access to critical resources. The defended space can range from a small nest site to extensive feeding or mating territories depending on the species, resource distribution, and ecological pressures. Animals use space in 3 major ways. 1. Home range, which is the area regularly traversed by an individual in search of food or mates but not actively defended. 2. Core area, a part of the home range used most intensively. And 3. Territory, a clearly Animals use space in three major ways. 1. Home range, which is the area regularly traversed by an individual in search of food or mates but not actively defended. 2. Core area, a part of the home range used most intensively. And 3. Territory, a clearly defined and actively defended portion of space. Not all animals defend territories, but many exhibit home range fidelity, returning to the same area repeatedly. The size and shape of territories are influenced by the distribution and abundance of resources, population density, mobility of the species, and cost-benefit trade-offs of defense. Territorial behavior is usually displayed when the benefits of exclusive access to resources outweigh the costs of defense, such as energy expenditure, time, and risk of injury. The economic defendability model explains that territoriality is favored when resources are sufficiently clumped and renewable, such that defending them yields a net fitness gain. If resources are too widely dispersed or unpredictable, it may not be feasible or advantageous to defend a territory. Territories can serve various functions. Feeding territories ensure access to food resources, mating territories secure reproductive opportunities, and nesting or breeding territories offer safe sites for raising offspring. For example, male red-winged blackbirds defend nesting territories that include prime vegetation cover for females to nest in, while hummingbirds establish feeding territories around rich flower patches. In some fish species like damselfish, algae-covered rocks are defended as feeding grounds. In lichen species, males defend small display territories within a communal lek to attract females, although they do not offer direct resources. Territoriality is maintained through visual, acoustic, chemical, or tactile signals rather than continuous physical aggression. Territorial animals often use ritualized displays, such as vocalizations in birds e.g., songbirds scent marking in mammals e.g., wolves, big cats, or visual posturing in lizards e.g., dewlap extension. These signals serve dual functions, advertising occupancy and deterring intruders. In some cases, direct aggression follows if an intruder challenges the resident's claim. Territorial contests often follow sequential assessment strategies and resource holding potential (RHP) frameworks. The RHP is a measure of an individual's capacity to win fights and maintain territories, based on attributes such as size, strength, energy reserves, and motivation. According to the bourgeois strategy, animals may resolve conflicts based on residency status. Residents typically win because they are more motivated and familiar with the area. This strategy reduces the frequency and cost of escalated fights. Territorial boundaries are usually well respected within stable social groups, but floating individuals, such as non-territorial males or dispersing juveniles, may attempt to intrude, take over, or sneak mating opportunities. In group living species, territories may be defended collectively. For instance, meerkats and chimpanzees patrol and defend group territories through coordinated activities, indicating that territoriality can extend beyond individuals to entire social units. Territoriality is not static. It is dynamic and context-dependent. Territories may expand, contract, shift, or be abandoned based on changes in resource availability, breeding status, dominance rank, or intra-group conflicts. In migratory species, territorial behavior may be limited to breeding seasons. In amphibians and reptiles, territorial claims may vary with temperature and breeding cycles. In some cases, temporal partitioning allows for shared use of space among individuals that avoid each other by being active at different times. Importantly, territoriality is not universal even within a species. Some individuals adopt non-territorial strategies, like sneaking or satellite behavior, especially in mating systems. This polymorphism is maintained through evolutionarily stable strategies where both territorial and non-territorial tactics yield comparable fitness outcomes over time. For example, in side-blotched lizards, different color morphs represent alternative strategies: aggressive territory holders, cooperative sharers, and sneaky invaders, each succeeding under specific social contexts. The evolution of territoriality is driven by natural selection for efficient space use, resource monopolization, and reproductive success. While it confers advantages such as increased access to mates, food, and shelter, it can also expose individuals to costs, like predation risk during display, injuries from territorial fights, and energetic burden. Therefore, territorial behavior is under strong selective pressure to be optimized rather than maximized. From an ecological perspective, territoriality has broader implications for population spacing patterns, density regulation, resource competition, and community dynamics. Territorial exclusion can lead to ideal free distributions, territory-based dominance hierarchies, and spatial segregation of age or sex classes. Thus, space use strategies like territoriality are integral to understanding animal behavior, evolution, and ecosystem function, an area repeatedly explored in CSIRNet questions. CSIRNet previous year questions: use of space and territoriality. CSIRNet previous year questions: use of space and territoriality. CSIRNet June 2017, Part C. In a bird species, males sing from specific perches, chase away intruders, and mate with females entering their area. Which of the following best describes this behavior? A home range behavior. B. Resource-based polygyny. C. Territory defense behavior. D. Kin selection behavior. Answer: C. Explanation: the male defends a fixed area with exclusive access to resources and mates. Territoriality. CSIRNet Deck 2019, Part B. Which of the following is likely to promote the evolution of territoriality? A widely dispersed and unpredictable resources. B. High cost of defending resources. C. Clumped and renewable resources. D. Uniform resource distribution and high population density. Answer: C. Explanation: clumped, predictable resources provide a defendable advantage. Economic defendability model. CSIRNet June 2015, Part C. In a species of lizards, territorial males use visual displays to repel rivals and attract mates. Subordinate males do not display but attempt sneaky matings. This is an example of a sexual dimorphism. B. Kin selection. C. Alternative mating strategies. D. Phenotypic plasticity. Answer: C. Explanation: coexistence of dominant territorial and sneaky non-territorial tactics reflects alternative strategies. Let me know if you're ready to continue with the next topic: mating systems and reproductive strategies, or any other area. I'm ready to provide it in the same ultra-detailed CSIRNet JRF format. You said, mating systems. ChatGPT said. Here is your CSIRNet JRF Life Sciences ultra-detailed, paragraph-based lecture on mating systems, written at the highest standard for cracking the CSIRNet JRF exam. It includes maximum word limit theory, no tables, and 3 previous year questions with answers and years mentioned, as requested. Mating systems, CSIRNet Life Sciences ultra-detailed lecture. A mating system is a pattern of mating behavior and reproductive association between individuals in a population. It defines how males and females pair or interact for reproduction and how resources, parental care, and sexual selection pressures shape these interactions. Mating systems are not just biological categories, they are complex outcomes of evolutionary trade-offs, ecological constraints, and sexual selection dynamics. The key factors influencing mating systems include operational sex ratio, parental investment, resource distribution, female distribution, and mate guarding opportunities. The classic classification of mating systems includes 4 major types: monogamy, polygyny, polyandry, and promiscuity or polygynandry. In monogamy, one male pairs with one female for a breeding season or a lifetime, often involving biparental care and strong pair bonds. This system is common in bird species like swans, albatrosses, and some primates like gibbons. Monogamy typically evolves when offspring survival depends on the care of both parents, or when females are widely dispersed, making it difficult for males to monopolize more than one mate. Polygyny, the most common mating system among mammals, involves one male mating with multiple females. Polygyny arises when males can monopolize access to multiple females, either directly or indirectly through control of resources. Polygyny is further classified based on how males gain access to females. In resource defense polygyny, males control territories rich in resources that attract females, e.g., red-winged blackbirds. In female defense polygyny, males directly guard groups of females, as seen in elephant seals and gorillas. In lek polygyny, males display in communal areas leks, and females choose among them based on display traits, e.g., sage grouse, mannequins. In these systems, sexual selection acts intensely on males, often resulting in sexual dimorphism and elaborate displays. Polyandry is less common and occurs when one female mates with multiple males. This system is often associated with sex role reversal, where males provide parental care. An example is the jacana bird, where females are territorial and compete for males who incubate eggs and care for young. Polyandry can be advantageous for females in terms of genetic diversity, increased fertility, and male assistance in rearing. Promiscuity or polygyny involves both sexes having multiple mating partners with no exclusive bonds. It is common in species where parental care is absent or shared communally. Examples include chimpanzees and bonobos. Promiscuity allows sperm competition, mate sampling, and cryptic female choice to operate as post-copulatory sexual selection mechanisms. These mating systems are influenced by the distribution and abundance of females, which in turn depend on resource availability. When females are clumped, it becomes economically beneficial for males to monopolize several mates, favoring polygyny. When females are evenly dispersed, monogamy is more likely. Ecological constraints such as predation risk, habitat type, and social structure also shape mating behavior. For instance, in high predation environments, parental cooperation monogamy may be essential for offspring survival. The Bateman principle and Treiber's theory of parental investment provide foundational explanations for mating systems. Bateman's experiments on fruit flies demonstrated that male reproductive success increases with the number of mates, while females reach reproductive success limits with fewer mates due to greater investment in gametes and often in offspring care. As a result, males compete, and females choose, a pattern that underlies the evolution of diverse mating systems. In some species, mating systems are flexible or plastic, varying with environmental or social conditions. For instance, Dunix prunella modularis exhibit all forms of mating systems, monogamy, polygyny, and polyandry, depending on food distribution and population density. This flexibility illustrates that mating systems are not rigid categories but context-dependent strategies that optimize reproductive success. Sexual selection, especially intersexual selection, mate choice, and intersexual competition, drives the evolution of mating systems. In liking species, females exert strong choice on male display traits, leading to exaggerated secondary sexual characteristics. In contrast, in polygynous species like deer or lions, male-male competition leads to large body size, weaponry-like antlers, and social dominance. The costs and benefits of different mating strategies are distributed asymmetrically between sexes. Males often invest in access to multiple mates, while females invest more in offspring quality and care. This results in the evolution of alternative mating strategies such as sneaking, satellite males, and sperm competition in males, and cryptic female choice, polyandry, and multiple paternity in females. Such adaptations can coexist within populations, maintained through evolutionarily stable strategies. Understanding mating systems also has implications in conservation biology, especially for species with complex social and reproductive systems. Knowledge of mating systems helps in designing captive breeding programs, maintaining genetic diversity, and predicting population dynamics under environmental change. Mating systems are a core topic in CSIR Net Life Sciences, and questions frequently test understanding of ecological drivers, types of mating systems, examples, and evolutionary consequences, particularly in the context of sexual selection and parental investment. CSIR Net previous year questions. Mating systems. CSIR Net Deck 2017, Part C. A bird species exhibits a mating system where females mate with multiple males, and males provide parental care. Which mating system best describes this behavior? A. Monogamy. B. Polygyny. C. Polyandry. D. Promiscuity. Answer C. Explanation. Female mates with multiple males, and males invest in parental care. This is classic polyandry. CSIR Net June 2018, Part B. Which condition is most likely to promote the evolution of monogamy in a species? A clumped distribution of females. B. High parental investment from both sexes. C. Male dominance hierarchies. D. Absence of parental care. Answer B. Explanation. When offspring survival depends on both parents, monogamy is favored evolutionarily. CSIR Net Deck 2019, Part C. In a liking species, which of the following is most likely to evolve? A. Male parental care. B. Equal mating success among males. C. Strong female choice and male-male competition. Female defense polygyny. Answer C. Explanation. Liking involves males displaying, females choosing, intense intersexual selection. Let me know when you're ready for the next lecture topic, parental investment and care, or anything else from your CSIR Net JRF Life Sciences syllabus. I'll continue in the same ultra-detailed format. You said. Parental investment and reproductive success. ChatGPT said. Here is your CSIR Net JRF Life Sciences ultra-detailed lecture on parental investment and reproductive success, structured for the highest level of preparation. It is fully paragraph-based, has no tables, includes maximum word coverage, and ends with 3 previous year CSIR Net questions with answers and years mentioned, as per your request. Parental investment and reproductive success, CSIR Net JRF Life Sciences lecture. Parental investment refers to any contribution made by a parent to the survival and fitness of their offspring that comes at a cost to the parent's ability to invest in other offspring. This concept, originally formalized by Robert Treiber in 1972, is foundational in understanding the evolution of mating systems, sexual selection, and life history strategies. The investment can be in the form of time, energy, protection, provisioning, or teaching behaviors and can occur before or after birth or hatching. Importantly, the key evolutionary trade-off here is between the quality of offspring and the quantity of offspring an individual can produce and successfully rear. The evolutionary basis of parental investment lies in maximizing an individual's inclusive fitness, which considers not only the survival of one's direct offspring but also kin that share genes. High parental investment typically increases the survival and future reproductive success of the offspring, but also limits the parent's future reproductive opportunities, either due to exhaustion of resources or lost mating opportunities. There is often an asymmetry in parental investment between the sexes, rooted in the biology of reproduction. Females typically invest more because of larger, energetically expensive gametes, eggs versus sperm gestation, and lactation in mammals. As a result, females are a limiting resource for male reproduction, leading to competition among males and choosiness among females, as per Bateman's principle and Treiber's theory of parental investment. Males may increase their reproductive success by mating with many females, while females benefit more by choosing high-quality mates or investing more in fewer offspring. Parental investment strategies vary widely among taxa. In many bird species, biparental care is common, as both male and female can contribute to feeding, guarding, and teaching chicks, thus improving offspring survival. In contrast, maternal care is more common in mammals due to internal gestation and lactation. Paternal care, although rare, can be found in certain fish like seahorses and sticklebacks, and amphibians, where external fertilization allows males to guard eggs and reduce the risk of cuckoldry. Alloparental care, where non-parents such as siblings or group members help raise offspring, also contributes to reproductive success and kin selection, especially in cooperative breeders like meerkats and some primates. A critical concept linked to parental investment is reproductive success, defined as the number of viable offspring an individual leaves in the next generation. Reproductive success is influenced not only by the number of offspring produced but also by how many survive to reproduce themselves. Thus, higher parental investment, especially under conditions of high predation or resource scarcity, can significantly enhance offspring survival and, consequently, reproductive success. The costs and benefits of parental care are subject to natural selection. If the benefit to the offspring in terms of increased survival and fitness outweighs the cost to the parent in terms of reduced future reproduction, then parental care is favored. In contrast, if the cost is too high or the benefit too low, e.g., in species with low offspring survival regardless of care, abandonment or minimal care strategies may evolve. Another major influence is certainty of parentage, especially in males. When males cannot be sure that the offspring are their own, they may withhold parental care to avoid wasting resources on unrelated young. In contrast, females usually have complete certainty of maternity, reinforcing their investment. This dynamic helps explain why male parental care is more common in species with external fertilization, where paternity is more assured, and less common in species with internal fertilization, where the potential for female multiple mating complicates paternity certainty. The level of parental investment also influences sexual dimorphism and mating systems. In species where males contribute little or no parental investment, sexual selection tends to favor traits that increase male mating success, like bright colors, large size, or elaborate displays. In contrast, where males invest heavily, sexual selection can act on both sexes or even reverse typical sex roles, as seen in polyandrous species like Jacanas, where females are larger and compete for males who care for the eggs. Parental investment also affects the evolution of life history traits, such as age of maturity, number of offspring, frequency of reproduction, and lifespan. Species that produce many offspring with little investment, e.g., many fishes, amphibians, invertebrates follow an R-selected strategy, while those that produce few offspring with high investment, e.g., elephants, humans follow a K-selected strategy. In recent studies, trade-offs between current and future reproduction have been shown in various species. For example, in birds, removing some eggs or chicks leaves parents to reduce future investment, supporting the idea of adaptive adjustment of care. Similarly, in species like burying beetles, parental care is adjusted according to the size and quality of the offspring or resource availability. Understanding the dynamics of parental investment is essential not only in evolutionary biology but also in conservation. In species with high parental investment, disruption of parental behavior due to environmental change or anthropogenic pressures, like poaching, habitat fragmentation, can lead to dramatic declines in reproductive success and population viability. CSIR Net previous year questions. Parental investment in reproductive success. CSIR Net Deck 2016, Part C. Which of the following best explains why females are generally more selective in choosing mates than males? A females have lower reproductive success than males. Females are typically larger than females. C. Females invest more energy into each offspring. D. Males care more for the young. Answer C. Explanation. Greater parental investment by females leads to increased selectivity in mate choice. CSIR Net June 2015, Part B. In species where external fertilization occurs, male parental care is more common because males are larger than females. B. Certainty of paternity is higher. C. Females produce more eggs. D. Males are under sexual selection. Answer B. Explanation. With external fertilization, males can see and guard eggs, increasing paternity certainty. CSIR Net Deck 2018, Part C. In a species of fish, males guard and then care for eggs. If paternity is experimentally reduced by allowing females to spawn with multiple males, what is a likely consequence? A. Males increase parental investment. B. Males show no change in behavior. C. Males reduce or abandon parental investment. D. Females increase egg production. Answer C. Explanation. Reduced certainty of paternity leads to reduced male investment in offspring. Let me know when you're ready for the next topic, aggressive behavior or any other from your CSIR Net Life Sciences syllabus. I will continue in this same in-depth format. You said, parental care, ChatGPT said. Here is your CSIR Net JRF Life Sciences Ultra Detailed Lecture on parental care, crafted for the highest standard of preparation. It is structured in a pure paragraph format, no tables, includes maximum word coverage, and ends with 3 previous year CSIR Net questions with answers and years mentioned, based strictly on previously asked areas and core exam themes. Parental care, CSIR Net JRF Life Sciences Lecture. Parental care refers to any behavioral and physiological activity performed by a parent that enhances the survival, development, or reproductive success of their offspring. This care can include feeding, guarding, grooming, incubation, teaching, or defending the young from predators or environmental stressors. It represents a key evolutionary adaptation that significantly improves offspring fitness but often comes at a cost to the parent in terms of energy expenditure, time, and reduced future reproductive opportunities. Parental care strategies are highly diverse across the animal kingdom and reflect the balance between offspring needs, environmental pressures, and life history strategies of the species. In many species, especially invertebrates and R strategists, there is minimal or no parental care. Organisms produce a large number of offspring and rely on sheer numbers to ensure survival. In contrast, K strategists such as birds and mammals produce fewer offspring but invest significantly in each one, improving the likelihood that each survives to maturity. One of the most influential theories explaining parental care is Triber's theory of parental investment, which posits that the sex investing more in parental care will be more selective in mate choice, while the less investing sex will compete for access to mates. Typically, females exhibit more parental care because they bear higher physiological investment during gestation and often lactation, especially in mammals. This sexual asymmetry leads to evolution of different behaviors, such as male-male competition and female choosiness. Parental care can be maternal, paternal, biparental, or even alloparental care by individuals other than the biological parents. In mammals, maternal care dominates due to internal fertilization and lactation, though in some species, e.g., lions, females may also cooperate in communal care. In birds, biparental care is common, as both sexes can contribute to nest building, incubation, and feeding. In contrast, in many amphibians and fish, paternal care is common, often due to external fertilization, where males can guard eggs late in the environment. The evolution of parental care involves multiple selective pressures. When the environment is unpredictable, predation risk is high, or offspring are born underdeveloped, ultrashell parental care becomes advantageous. In contrast, when offspring are precocial able to survive independently at birth, and mortality is high regardless of care, natural selection may not favor intensive care. Different forms of care have evolved, including brooding, e.g., in birds and frogs feeding, regurgitation or milk guarding, seen in fish and insects, and nest defense. Some reptiles like pythons coil around their eggs, increasing hatchling success through thermoregulation. In insects like burying beetles, both parents may prepare a food source for larvae and defend it from rivals. In amphibians like poison dart frogs, adults may carry tadpoles on their backs to safe water sources. Parental care is often flexible and influenced by cost-benefit trade-offs. If offspring are likely to survive without assistance, or if the parent's future reproductive potential is jeopardized, care may be minimized. The concept of reproductive value becomes essential in such decisions. Parents may abandon a low-quality brood to invest in future, more promising reproductive attempts. In some cases, parents may even cannibalize offspring, a behavior observed in some fishes and amphibians, which may recycle nutrients or remove unfit young. Parental care is tightly linked to reproductive success. Species with high parental care generally show increased survival rates for offspring, enhanced growth, better learning, and greater chances of reproductive success in the next generation. This explains why many species evolve complex social behaviors around parenting. In social insects like bees and ants, sterile workers invest in the queen's offspring, kin selection demonstrating extreme forms of parental and alloparental care. Moreover, certainty of parentage influences the evolution of care. In internally fertilizing species, males often avoid care due to uncertainty over paternity, whereas in externally fertilizing species, such as many fish, males frequently provide care because they are more confident the eggs are their own. This links closely to sexual selection. Male care is more likely to evolve when males can ensure paternity, or when female choice favors caring males. Importantly, parental care has been shaped not only by natural selection but also by sexual selection and social ecology. For instance, in species with female defense polygyny, males guard and care for offspring in return for access to multiple females. In monogamous systems, cooperative parenting is favored and linked to higher offspring survival. In some birds, the lack of paternal care results in increased female workload and potential reproductive trade-offs. Parental care is also influenced by environmental conditions. Resource availability, predation risk, parasite pressure, and social group structure can all impact the intensity, type, and duration of care. For example, in fluctuating environments, plasticity in parental behavior, adjusting care based on conditions or offspring demand, may offer an evolutionary advantage. In cooperative breeding systems, e.g., meerkats, non-breeding individuals, helpers contribute to raising young. In cooperative breeding systems, e.g., meerkats, non-breeding individuals, helpers contribute to raising young, a form of kin-selective parental care. In human evolution, parental care has taken on unique complexities, involving extended juvenile dependency, teaching, provisioning, and emotional bonding. Human parenting includes not only direct biological investment but also sociocultural aspects that shape the next generation's fitness in both biological and cultural dimensions. CSIRNet Previous Year Questions. Parental Care CSIRNet Deck 2013, Part C. In a species of fish, males are known to guard fertilized eggs until hatching. Which factor best explains the evolution of this paternal care? A higher fecundity in males. B. External fertilization and certainty of paternity. C. Male hormonal cycles. D. Female egg laying behavior. Answer B. Explanation. Males are more likely to provide care when they are confident the offspring are theirs, as in external fertilization. CSIRNet June 2017, Part B. Which of the following conditions is least likely to favor the evolution of parental care? A high predation risk to offspring. B. High certainty of parentage. C. Short lifespan of adults. D. High value of each offspring due to small clutch size. Answer C. Explanation. A short adult lifespan may limit the benefit of investing heavily in each reproductive attempt. CSIRNet Deck 2019, Part C. A bird species lays 3 eggs. One parent is experimentally removed after laying. Which of the following is the likely outcome? A remaining parent compensates and all chicks survive. B. Remaining parent reduces care and only one chick survives. C. All chicks die due to predation. D. The parent shifts to caring for other offspring elsewhere. Answer B. Explanation. In many biparental systems, removing one parent reduces total care and decreases chick survival. Let me know when you're ready for the next topic. Aggressive behavior. CSIRNet Previous Year Questions. Parental Care CSIRNet Deck 2013, Part C. In a species of fish, males are known to guard fertilized eggs until hatching. Which factor best explains the evolution of this paternal care? A higher fecundity in males. B. External fertilization and certainty of paternity. C. Male hormonal cycles. D. Female egg laying behavior. Answer B. Explanation. Males are more likely to provide care when they are confident the offspring are theirs, as in external fertilization. CSIRNet June 2017, Part B. Which of the following conditions is least likely to favor the evolution of parental care? A high predation risk to offspring. B. High certainty of parentage. C. Short lifespan of adults. D. High value of each offspring due to small clutch size. Answer C. Explanation. A short adult lifespan may limit the benefit of investing heavily in each reproductive attempt. CSIRNet Deck 2019, Part C. A bird species lays 3 eggs. One parent is experimentally removed after laying. Which of the following is the likely outcome? A remaining parent compensates and all chicks survive. B. Remaining parent reduces care and only one chick survives. C. All chicks die due to predation. D. The parent shifts to caring for other offspring elsewhere. Answer B. Explanation. In many biparental systems, removing one parent reduces total care and decreases chick survival. Let me know when you're ready for the next topic. Aggressive behavior or any other. I'll continue with the same thorough, paragraph-based, CSIRNet JRF level notes tailored to help you crack the exam with just this material. You said. Aggressive behavior. ChatGPT said. Here is your CSIRNet JRF Life Sciences Ultra Detailed Lecture on Aggressive Behavior, written at the highest level of depth and specificity for the exam. It includes no tables, is in dense paragraph format, covers maximum word limit, and is based strictly on frequently asked and expected areas in CSIRNet, especially Part B and C. The lecture ends with 3 solved CSIRNet previous year questions with year and answer. Aggressive behavior, CSIRNet JRF Life Sciences Lecture. Aggressive behavior is a fundamental aspect of animal behavior, defined as any act or threat of action by one individual that potentially harms another individual of the same or different species. It plays a central role in the regulation of social interactions, access to resources, mating opportunities, territory defense. Aggression is considered a behavioral adaptation shaped by natural and sexual selection because, when expressed appropriately, it increases an individual's chances of survival and reproductive success. However, aggression carries a cost, risk of injury, energy expenditure, and sometimes social exclusion, so its expression is often regulated through evolved mechanisms such as dominance hierarchies, signaling, and ritualized combat. From an ethological perspective, aggression can be classified into several types: territorial aggression to defend space, maternal aggression to protect offspring, sexual aggression in mating contexts, dominance aggression in social hierarchies, and predatory aggression involving hunting. In many species, especially vertebrates, aggression is not random but follows specific patterns mediated by hormones, neural circuits, and environmental stimuli. For example, testosterone has been shown to increase aggressive tendencies in males of many vertebrates, whereas serotonin often plays an inhibitory role. The amygdala, hypothalamus, and periaqueductal gray are key brain regions involved in controlling aggressive behaviors. A significant component of aggression is the ritualization of conflict. Instead of direct physical combat, which can be fatal, many species have evolved stereotyped behaviors that signal intent and strength. These displays, such as roaring in lions, antler display in deer, or fin flaring in fish, help settle disputes without injury. This principle was described by ethologist Conrad Lorenz, who emphasized that interspecific aggression within the same species is often governed by rules of engagement and appeasement behaviors that reduce the likelihood of harm. Aggressive encounters may escalate step by step, allowing the weaker individual to retreat before injury occurs. This evolutionarily stable strategy conserves energy and prevents self-damage. Aggression and dominance hierarchies are tightly linked. In many group-living species, individuals establish and maintain rank through aggressive interactions. Once a hierarchy is formed, repeated aggression is minimized, as subordinates avoid challenging dominance. This reduces the cost of constant fighting and maintains group stability. For example, in primates such as baboons, alpha individuals often monopolize resources and mates, while subordinates engage in submissive displays to avoid confrontation. In social insects like ants and bees, aggression is used to maintain reproductive roles and task allocation. Aggression is also influenced by resource holding potential (RHP) and individuals' ability to win contests. RHP depends on factors such as size, strength, health, experience, and motivation. Individuals with higher RHP tend to initiate and win aggressive encounters. However, aggression is not only a function of physical capacity but also decision-making based on the value of the resource value theory. An animal is more likely to escalate aggression when the contested resource is highly valuable, such as a mate or nesting site. From an evolutionary standpoint, aggression must be adaptive. The cost-benefit model of aggression posits that aggressive behavior evolves only when the benefits, e.g., access to mates, territory, food outweigh the costs, e.g., energy loss, injury, retaliation. In some species, aggression has evolved as part of sexual selection. For instance, male-male combat in elephant seals determines reproductive access to harems, and only dominant male sire offspring. Similarly, in birds like red-winged blackbirds, territorial aggression increases mating opportunities and nest success. Kin selection theory helps explain the restraint often seen in aggression toward relatives. Inclusive fitness benefits discourage aggression among kin unless reproductive competition is extreme. For example, in naked mole rats, subordinate workers rarely show aggression toward kin because cooperative care of relatives increases indirect fitness. Conversely, when unrelated individuals compete for breeding rights or limited resources, aggression can be intense and sustained. Aggression is also shaped by ecological and social environments. In high-density populations, aggression may increase due to resource scarcity and social stress. In contrast, stable groups with well-established hierarchies often show reduced aggression. Environmental triggers such as seasonal changes, territory intrusion, or mate competition often elevate aggression levels temporarily. For instance, male songbirds exhibit peak aggression during the breeding season when defending nests. Interestingly, some species show sex-specific aggression patterns, with males more aggressive in mating contexts and females displaying maternal aggression. In humans and some primates, social factors, learning, and experience modulate aggression beyond instinctive drives. Culture, past victories or defeats, winner-loser effects, and hormonal states influence the likelihood of initiating aggression. Experimental models using rodents have revealed that repeated defeat in social aggression can induce stress and social withdrawal, demonstrating the neurobiological impact of aggression beyond immediate behavioral outcomes. Finally, aggression is not universally beneficial. Natural selection can favor cooperation over aggression when group benefits outweigh individual competition, as seen in social species like wolves, dolphins, and cooperative breeders. In such systems, aggression is regulated through social bonding, affiliative behaviors, and indirect reciprocity. In sum, aggressive behavior is a multifaceted phenomenon that balances competition with cooperation, cost with benefit, and instinct with experience. Its expression is under tight evolutionary, ecological, hormonal, and neural control, making it a central theme in behavioral biology and animal ecology, frequently explored in CSIRNet questions due to its integrative nature. CSIRNet previous year questions. Aggressive behavior. CSIRNet Deck 2014, Part C. Two male birds engage in a prolonged display of songs and postures without physical fighting. This behavior is best explained by a mating cooperation. B. Alloparental care. C. Ritualized aggression to avoid costly fights. D. Predator distraction. Answer C. Two male birds engage in a prolonged display of songs and postures without physical fighting. This behavior is best explained by a mating cooperation. B. Alloparental care. C. Ritualized aggression to avoid costly fights. D. Predator distraction. Answer C. Explanation. The behavior represents ritualized conflict, a common evolutionary strategy to resolve disputes without injury. CSIRNet June 2016, Part B. In species with dominance hierarchies, aggression is minimized after ranks are established. This benefits the group by a promoting indiscriminate mating. B. Increasing overall group aggression. C. Reducing energy loss from constant fighting. D. Eliminating weaker individuals. Answer C. Explanation. Established hierarchies reduce repeated aggression, conserving energy and promoting group stability. CSIRNet Deck 2018, Part C. A researcher finds that male fish are more aggressive during mating season and only in the presence of other males. This suggests aggression is a non-adaptive and accidental. B. Socially triggered and reproductive in function. C. Based on kin selection. D. Resulting from resource abundance. Answer B. Explanation. Aggression is context dependent and related to competition for mates, a common evolutionary pattern. Let me know when you're ready for the next topic: habitat selection, foraging optimality, migration, orientation and navigation, or any other CSIRNet JRF life sciences subject. Each will be covered in the same high-quality, exam-oriented, paragraph-based format. You said. Habitat selection and optimality in foraging. ChatGPT said. Here is your CSIRNet JRF Life Sciences high-level lecture on habitat selection and optimality in foraging, written in dense, paragraph-based format with no tables, maximum elaboration, and focused on CSIRNet syllabus and question trends. The content integrates base to advanced level theory and ends with 3 previous year CSIRNet questions with answers in here. Habitat selection and optimality in foraging, CSIRNet Life Sciences lecture. Habitat selection refers to the behavioral process by which organisms choose specific environments to live, forage, breed, and shelter, based on cues that reflect the suitability of a habitat for survival and reproductive success. This selection is not random. Rather, it is shaped by both evolutionary history and ecological optimization, meaning animals evolve preferences that increase their fitness by maximizing the benefits, e.g., food, mates, safety, while minimizing the costs, e.g., predation risk, competition, harsh climate. From a behavioral ecology perspective, habitat choice is viewed as an adaptive decision-making process in which individuals assess environmental features, use past experience, or even copy others' choices, conspecific attraction, depending on the context. A central concept in habitat selection is the ideal free distribution (IFD) model, which postulates that individuals will distribute themselves among different habitat patches in proportion to the availability of resources. In an ideal condition, animals are assumed to have perfect knowledge of habitat quality, and under free movement, they can go wherever they choose. According to IFD, as more individuals occupy a high-quality patch, its per capita benefit decreases due to increased competition, thereby making lower-quality patches more attractive. Eventually, animals settle in a way that equalizes their payoff across habitats. However, in real systems, constraints such as territoriality, social hierarchy, predation risk, or limited information make the distribution non-ideal and non-free, leading to deviations from the IFD predictions. Habitat selection decisions are deeply interconnected with foraging behavior, especially under conditions where food is patchily distributed. This brings in the theory of optimal foraging, which is one of the most testable and widely applied concepts in behavioral ecology. The optimal foraging theory (OFT) is based on the premise that animals are selected to forage in such a way that they maximize energy intake per unit time, while minimizing energy expenditure and risk. The classical framework of OFT is the energy maximization hypothesis, where foraging decisions, e.g., which food to eat, how long to stay in a patch, which patches to visit, are shaped by the trade-offs between gain and cost. A key model within OFT is the patch use model, particularly the marginal value theorem (MVT) proposed by Eric Charnov. It describes the optimal time an animal should spend in a food patch before moving to the next. According to MVT, an animal should leave a patch when the instantaneous rate of food intake drops below the average rate in the environment. For instance, a bird feeding in a bush should stop foraging there and move to the next when the food yield starts diminishing and becomes less than what is expected elsewhere, taking into account the travel time. This model helps explain patch departure decisions, especially in species like bumblebees, rodents, or primates that exploit food clumps. The diet choice model, another important component of OFT, explains how animals decide which prey or food items to include in their diet. It predicts that animals should specialize on the most profitable prey when such prey is abundant and broad in their diet when it is scarce. The profitability of a prey is determined by the ratio of its energy content to the handling time. For example, a predator may ignore slow-moving but low-calorie insects if high-calorie prey is sufficiently available. However, animals do not always behave as perfect optimizers. Their foraging behavior is often constrained by predator risk, nutrient balancing, toxicity avoidance, competition, and learning limitations. These factors lead to state-dependent foraging, where hunger level, energy reserves, or reproductive status influence choices. For example, a starving animal may take greater risks than a well-fed one. Additionally, risk-sensitive foraging predicts that animals sometimes prefer patches with more predictable, though smaller food rewards over patches with higher but variable rewards when the variance in resource availability may result in starvation. Habitat selection and foraging also exhibit strong coevolution with sensory and cognitive traits. Animals evolve specific sensory abilities to detect suitable habitats and profitable food items, such as olfactory cues in moths for host plant selection or echolocation in bats. Cognitive traits such as memory, learning, and spatial mapping are crucial for locating rich foraging sites and avoiding depleted patches. Migratory species often rely on these abilities to return to productive habitats across seasons, while territorial animals may show site fidelity, returning to the same successful locations. In some cases, animals use public information or social learning to optimize habitat and foraging choices. For example, honeybee foragers communicate patch quality through the waggle dance, allowing others to exploit rich patches. Similarly, birds and fishes make copy foraging decisions of conspecifics when unsure about the environment. This copy when uncertain strategy is adapted in fluctuating environments where individual trial and error learning is costly. From an evolutionary perspective, both habitat selection and foraging behavior under strong natural selection, as they directly impact an organism's survival and reproduction. Errors in habitat choice can lead to ecological traps, where animals are lured into low-quality habitats due to misleading cues, e.g., choosing artificial surfaces that reflect polarized light as water. Understanding these behaviors also has conservation implications, especially in the design of wildlife corridors, habitat restoration, and reserve design, where promoting natural habitat selection patterns can improve population viability. In CSIRNet, questions often test conceptual clarity of IFD, MBT, diet choice, risk-sensitive foraging, and behavioral trade-offs. These theories provide a predictive framework and remain cornerstones in behavioral and evolutionary ecology. Habitat selection refers to the behavioral process by which organisms choose specific environments to live, forage, breed, and shelter, based on cues that reflect the suitability of a habitat for survival and reproductive success. This selection is not random, rather, it is shaped by both evolutionary history and ecological optimization, meaning animals evolve preferences that increase their fitness by maximizing the benefits, e.g., food, mates, safety, while minimizing the costs, e.g., predation risk, competition, harsh climate. From a behavioral ecology perspective, habitat choice is viewed as an adaptive decision-making process in which individuals assess environmental features, use past experience, or even copy others' choices conspecific attraction depending on the context. A central concept in habitat selection is the ideal free distribution (IFD) model, which postulates that individuals will distribute themselves among different habitat patches in proportion to the availability of resources. In an ideal condition, animals are assumed to have perfect knowledge of habitat quality, and under free movement, they can go wherever they choose. According to IFD, as more individuals occupy a high-quality patch, its per capita benefit decreases due to increased competition, thereby making lower-quality patches more attractive. Eventually, animals settle in a way that equalizes their payoff across habitats. However, in real systems, constraints such as territoriality, social hierarchy, predation risk, or limited information make the distribution non-ideal and non-free, leading to deviations from the IFD predictions. Habitat selection decisions are deeply interconnected with foraging behavior, especially under conditions where food is patchily distributed. This brings in the theory of optimal foraging, which is one of the most testable and widely applied concepts in behavioral ecology. The optimal foraging theory (OPT) is based on the premise that animals are selected to forage in such a way that they maximize energy intake per unit time, while minimizing energy expenditure and risk. The classical framework of OPT is the energy maximization hypothesis, where foraging decisions, e.g., which food to eat, how long to stay in a patch, which patches to visit, are shaped by the trade-offs between gain and cost. A key model within OPT is the patch use model, particularly the marginal value theorem (MVT) proposed by Eric Charnov. It describes the optimal time an animal should spend in a food patch before moving to the next. According to MVT, an animal should leave a patch when the instantaneous rate of food intake drops below the average rate in the environment. For instance, a bird feeding in a bush should stop foraging there and move to the next when the food yield starts diminishing and becomes less than what is expected elsewhere, taking into account the travel time. This model helps explain patch departure decisions, especially in species like bumblebees, rodents, or primates that exploit food clumps. The diet choice model, another important component of OPT, explains how animals decide which prey or food items to include in their diet. It predicts that animals should specialize on the most profitable prey when such prey is abundant and broad in their diet when it is scarce. The profitability of a prey is determined by the ratio of its energy content to the handling time. For example, a predator may ignore slow-moving but low-calorie insects if high-calorie prey is sufficiently available. However, animals do not always behave as perfect optimizers. Their foraging behavior is often constrained by predator risk, nutrient balancing, toxicity avoidance, competition, and learning limitations. These factors lead to state-dependent foraging, where hunger level, energy reserves, or reproductive status influence choices. For example, a starving animal may take greater risks than a well-fed one. Additionally, risk-sensitive foraging predicts that animals sometimes prefer patches with more predictable, though smaller food rewards over patches with higher but variable rewards when the variance in resource availability may result in starvation. Habitat selection and foraging also exhibit strong coevolution with sensory and cognitive traits. Animals evolve specific sensory abilities to detect suitable habitats and profitable food items, such as olfactory cues in moths for host plant selection or echolocation in bats. Cognitive traits such as memory, learning, and spatial mapping are crucial for locating rich foraging sites and avoiding depleted patches. Migratory species often rely on these abilities to return to productive habitats across seasons, while territorial animals may show site fidelity, returning to the same successful locations. In some cases, animals use public information or social learning to optimize habitat and foraging choices. For example, honeybee foragers communicate patch quality through the waggle dance, allowing others to exploit rich patches. Similarly, birds and fishes may copy foraging decisions of conspecifics when unsure about the environment. This copy when uncertain strategy is adapted in fluctuating environments where individual trial and error learning is costly. From an evolutionary perspective, both habitat selection and foraging behavior under strong natural selection, as they directly impact an organism's survival and reproduction. Errors in habitat choice can lead to ecological traps, where animals are lured into low-quality habitats due to misleading cues, e.g., choosing artificial surfaces that reflect polarized light as water. Understanding these behaviors also has conservation implications, especially in the design of wildlife corridors, habitat restoration, and reserve design, where promoting natural habitat selection patterns can improve population viability. In CSIRNet, questions often test conceptual clarity of IFD, MBT, diet choice, risk-sensitive foraging, and behavioral trade-offs. These theories provide a predictive framework and remain cornerstones in behavioral and evolutionary ecology. CSIRNet previous year questions. Habitat selection and optimal foraging. CSIRNet June 2017, Part C. A bird species forages in patches with different food densities. According to the marginal value theorem, the bird should leave a patch when the patch becomes empty. B. The intake rate falls below the average rate in the habitat. C. Another bird arrives in the patch. D. The prey become too small to detect. Answer B. Explanation. MBT predicts patch departure when marginal gain drops below the environment's average. CSIRNet Deck 2019, Part provide a predictive framework and remain cornerstones in behavioral and evolutionary ecology. CSIRNet previous year questions. Habitat selection and optimal foraging. CSIRNet June 2017. Part C. A species forages in patches with different food densities. According to the marginal value theorem, the bird should leave a patch when the patch becomes empty. B. The intake rate falls below the average rate in the habitat. C. Another bird arrives in the patch. D. The prey becomes too small to detect. Answer. B. Explanation. MBT predicts patch departure when marginal gain drops below the environment's average. CSIRNet Deck 2019. Part B. Which of the following best explains the ideal free distribution model? All individuals have equal access to habitats but prefer social habitats. B. Individuals distribute proportionally to resource availability if they have perfect knowledge and no movement constraints. C. Only dominant individuals access the best patches. All individuals have equal access to habitats but prefer social habitats. B. Individuals distribute proportionally to resource availability if they have perfect knowledge and no movement constraints. C. Only dominant individuals access the best patches. Habitat selection and optimality in foraging. ChatGPT said. Here is your CSIRNet JRF Life Sciences high-level lecture on habitat selection and optimality in foraging, written in dense, paragraph-based format with note tables, maximum elaboration, and focused on CSIRNet syllabus and question trends. The content integrates base to advanced level theory and ends with 3 previous year CSIRNet questions with answers in year. Habitat selection and optimality in foraging. CSIRNet Life Sciences lecture. Habitat selection refers to the behavioral process by which organisms choose specific environments to live, forage, breed, and shelter, based on cues that reflect the suitability of a habitat for survival and reproductive success. This selection is not random. Rather, it is shaped by both evolutionary history and ecological optimization, meaning animals evolve preferences that increase their fitness by maximizing the benefits, e.g., food, mates, safety, while minimizing the costs, e.g., predation risk, competition, harsh climate. From a behavioral ecology perspective, habitat choice is viewed as an adaptive decision-making process in which individuals assess environmental features, use past experience, or even copy others' choices, conspecific attraction, depending on the context. A central concept in habitat selection is the ideal free distribution (IFD) model, which postulates that individuals will distribute themselves among different habitat patches in proportion to the availability of resources. In an ideal condition, animals are assumed to have perfect knowledge of habitat quality, and under free movement, they can go wherever they choose. According to IFD, as more individuals occupy a high-quality patch, its per capita benefit decreases due to increased competition, thereby making lower-quality patches more attractive. Eventually, animals settle in a way that equalizes their payoff across habitats. However, in real systems, constraints such as territoriality, social hierarchy, predation risk, or limited information make the distribution non-ideal and non-free, leading to deviations from the IFD predictions. Habitat selection decisions are deeply interconnected with foraging behavior, especially under conditions where food is patchily distributed. This brings in the theory of optimal foraging, which is one of the most testable and widely applied concepts in behavioral ecology. The optimal foraging theory (OFT) is based on the premise that animals are selected to forage in such a way that they maximize energy intake per unit time, while minimizing energy expenditure and risk. The classical framework of OFT is the energy maximization hypothesis, where foraging decisions, e.g., which food to eat, how long to stay in a patch, which patches to visit, are shaped by the trade-offs between gain and cost. A key model within OFT is the patch use model, particularly the marginal value theorem (MBT) proposed by Eric Charnov. It describes the optimal time an animal should spend in a food patch before moving to the next. According to MBT, an animal should leave a patch when the instantaneous rate of food intake drops below the average rate in the environment. For instance, a bird feeding in a bush should stop foraging there and move to the next when the food yield starts diminishing and becomes less than what is expected elsewhere, taking into account the travel time. This model helps explain patch departure decisions, especially in species like bumblebees, rodents, or primates that exploit food clumps. The diet choice model, another important component of OFT, explains how animals decide which prey or food items to include in their diet. It predicts that animals should specialize on the most profitable prey when such prey is abundant and broad in their diet when it is scarce. The profitability of a prey is determined by the ratio of its energy content to the handling time. For example, a predator may ignore slow-moving but low-calorie insects if high-calorie prey is sufficiently available. However, animals do not always behave as perfect optimizers. Their foraging behavior is often constrained by predator risk, nutrient balancing, toxicity avoidance, competition, and learning limitations. These factors lead to state-dependent foraging, where hunger level, energy reserves, or reproductive status influence choices. For example, a starving animal may take greater risks than a well-fed one. Additionally, risk-sensitive foraging predicts that animals sometimes prefer patches with more predictable, though smaller food rewards over patches with higher but variable rewards when the variance in resource availability may result in starvation. Habitat selection and foraging also exhibit strong coevolution with sensory and cognitive traits. Animals evolve specific sensory abilities to detect suitable habitats and profitable food items, such as olfactory cues in moths for host plant selection or echolocation in bats. Cognitive traits such as memory, learning, and spatial mapping are crucial for locating rich foraging sites and avoiding depleted patches. Migratory species often rely on these abilities to return to productive habitats across seasons, while territorial animals may show site fidelity, returning to the same successful locations. In some cases, animals use public information or social learning to optimize habitat and foraging choices. For example, honeybee foragers communicate patch quality through the waggle dance, allowing others to exploit rich patches. Similarly, birds and fishes may copy foraging decisions of conspecifics when unsure about the environment. This copy when uncertain strategy is adapted in fluctuating environments where individual trial and error learning is costly. From an evolutionary perspective, both habitat selection and foraging behavior are under strong natural selection, as they directly impact an organism's survival and reproduction. Errors in habitat choice can lead to ecological traps, where animals are lured into low-quality habitats due to misleading cues, e.g., choosing artificial surfaces that reflect polarized light as water. Understanding these behaviors also has conservation implications, especially in the design of wildlife corridors, habitat restoration, and reserve design, where promoting natural habitat selection patterns can improve population viability. In CSIRNet, questions often test conceptual clarity of IFD, MBT, diet choice, risk-sensitive foraging, and behavioral trade-offs. These theories provide a predictive framework and remain cornerstones in behavioral and evolutionary ecology. CSIRNet previous year questions. Habitat selection and optimal foraging. CSIRNet June 2017. Part C. A bird species forages in patches with different food densities. According to the marginal value theorem, the bird should leave a patch when the patch becomes empty. B. The intake rate falls below the average rate in the habitat. C. Another bird arrives in the patch. D. The prey becomes too small to detect. Answer. B. Explanation. MBT predicts patch departure when marginal gain drops below the environment's average. CSIRNet Deck 2019. Part B. Which of the following best explains the ideal free distribution model? All individuals have equal access to habitats but prefer social habitats. B. Individuals distribute proportionally to resource availability if they have perfect knowledge and no movement constraints. C. Only dominant individuals access the best patches. D. Individuals always avoid competition. Answer. B. Explanation. IFD assumes individuals with perfect knowledge distribute according to habitat profitability. CSIRNet June 2021. Part C. An animal is given a choice between two food sources. One has low but consistent return. The other has high but variable return. If the animal is well fed, it is more likely to choose the risky, variable option. B. The consistent, low reward option. C. Both randomly. D. Neither, since it is satiated. Answer. B. Explanation. Risk-sensitive foraging predicts risk-averse behavior when energy needs are met. Let me know when you're ready for the next lecture on migration, orientation and navigation, or any other CSIRNet life sciences topic. Each will be structured in this same high-quality format, tuned specifically to your exam prep. You said, migration, ChatGPT said. Here is your CSIR-Net JRF Life Sciences high-quality lecture on migration, written in dense, paragraph-based style, with maximum word limit, no tables, and crafted specifically for CSIR-Net JRF level. It includes base to advanced level concepts, areas from previous CSIR questions, and 3 past year questions with answers in year at the end. Migration, CSIR-Net Life Sciences lecture. Migration is a long-distance, seasonal movement of animals from one habitat to another, typically associated with changes in environmental conditions such as temperature, resource availability, or reproductive cycles. It is one of the most dramatic and ecologically significant behavioral phenomena observed in the animal kingdom. Migration differs fundamentally from other types of movement like dispersal or nomadism due to its predictability, periodicity, and directionality, and often involves return journeys, round-trip migration across generations. The behavior is evolutionarily shaped by natural selection to enhance survival and reproductive success under fluctuating environmental conditions. The ultimate cause of migration lies in the adaptive advantage it provides. By migrating, organisms can escape harsh climates, reduce competition, exploit seasonally abundant resources, or find optimal breeding or nesting sites. For example, many birds migrate from temperate regions to the tropics to escape winter food scarcity and return to exploit rich breeding grounds. Similarly, marine species like whales migrate between feeding and calving grounds, and insects like monarch butterflies follow well-established transcontinental routes to maintain population survival. Proximate mechanisms underlying migration are driven by complex physiological and environmental cues. These include changes in day length, photoperiod, temperature shifts, food availability, hormonal changes, and genetic programming. Photoperiod is one of the most consistent cues triggering migratory behavior, especially in birds, where increasing day length in spring stimulates hormonal changes that prepare them for migration. The endocrine system, particularly melatonin and gonadotropins, plays a central role in initiating migratory readiness, such as fat deposition, muscle development, and increased restlessness zoogonorrhea in captive birds. Insects like locusts show migratory morphs triggered by population density, indicating that migration can also be induced by social or environmental stress. From a neuroethological perspective, internal biological clocks synchronize with environmental rhythms to time migration precisely. These circadian and circannual clocks are genetically encoded and modulated by environmental cues. In birds, migratory timing can be shifted experimentally by altering photoperiod, which shifts their endogenous rhythms. In monarch butterflies, migration timing is linked with photoperiodic control of juvenile hormone synthesis, ensuring seasonal flight occurs at the correct stage of development. Navigation and orientation, critical components of migration, are accomplished by integrating multiple sensory modalities including visual landmarks, olfactory cues, the position of the sun or stars, celestial cues, Earth's magnetic field, magnetoreception, and in some cases, polarized light. For instance, pigeons can return home over long distances using both magnetic cues and visual maps, while salmon use olfactory imprinting to return to natal rivers after years in the ocean. Sea turtles also demonstrate remarkable navigational abilities using the Earth's magnetic field to locate nesting beaches thousands of kilometers away. Insect migration, like that of monarch butterflies or dragonflies, often uses solar navigation and polarized light for orientation, guided by internal circadian clocks that adjust the sun's position throughout the day. Genetic and epigenetic basis of migration has been studied extensively in birds and insects. For example, in black-capped warblers, Sylvia tripicilla, migratory direction and duration are genetically determined, with hybrids showing intermediate routes. Recent research also suggests that epigenetic mechanisms like DNA methylation may regulate gene expression associated with migratory readiness and behavior. Migration imposes considerable energetic costs, and animals undergo physiological preparations such as hyperphagia, excessive eating, fat accumulation, muscle hypertrophy, and changes in metabolism. Birds may double their body weight before migration, storing fat as high-energy fuel. Some species exhibit torpor or rest phases to conserve energy en route, while others use aerodynamic formations, like V-shaped flight and geese, to reduce drag and save energy. The choice of migratory strategy, long non-stop flights, step migration, or loop migration, is influenced by species ecology, body size, resource availability, and predation risk. Migration also has profound ecological and evolutionary consequences, influencing gene flow between populations, the spread of diseases, predator-prey dynamics, and ecosystem nutrient cycling. Migrants often act as vectors for pathogens, e.g., avian flu in birds, and their mass arrivals or departures can shift food web dynamics. Additionally, climate change is significantly altering migratory patterns, with shifts in timing, phenology, routes, and destinations, leading to mismatch with resource availability and reduced reproductive success in some species. These changes have major conservation implications, especially for long-distance migrants whose journeys depend on multiple geographically distant stopover sites. From a conservation perspective, migration poses challenges due to habitat fragmentation, light pollution, urbanization, hunting, and climate disruptions. Conservation strategies require international cooperation, e.g., the Convention on Migratory Species and protection of flyways, corridors, and stopover habitats. Satellite telemetry and geolocators are now used extensively to study migration and inform conservation policies. Migration is a high-yield topic in CSIR-Net Life Sciences, with questions frequently focusing on navigation strategies, physiological mechanisms, orientation cues, energy economics, and evolutionary significance. Understanding the integration of proximate cues with ultimate evolutionary benefits is crucial to answer behavioral ecology questions effectively. CSIR-Net Previous Year Questions. Migration. CSIR-Net Deck 2018, Part C. Which of the following cues is least likely to be used by migratory birds for long-distance navigation? Earth's magnetic field. B. Visual landmarks. C. Sound cues from other birds. D. Celestial cues, stars and sun. Answer. C. Explanation. Sound is not used for long-distance navigation. Magnetic and celestial cues are key. CSIR. CSIR-Net June 2016. Part B. Zoogonorrhea observed in birds is associated with a sleeping patterns during migration. B. Increased aggression during breeding season. C. Nocturnal restlessness prior to migration. D. Territorial defense in migratory areas. Answer. C. Explanation. Zoogonorrhea is migratory restlessness, a pre-migratory behavior. CSIR-Net Deck 2020. Part C. Which of the following animals uses olfactory imprinting for navigation during migration? A. Monarch butterflies. B. Arctic tern. C. Salmon. D. Wildebeest. Answer. C. Explanation. Salmon use olfactory cues to return to their natal streams. Let me know when you're ready for the next lecture, orientation and navigation, biological rhythms, or any other key CSIR-Net Life Sciences topic. I'll continue with the same standard, full detail, paragraph style, and previous year questions included. You said, orientation. ChatGPT said. Here is your CSIR-Net JRF Life Sciences high-quality lecture on orientation, written in an elaborate, paragraph-based format with no tables, tailored precisely for the CSIR-Net JRF level based on previous year question trends, from basic to advanced concepts. This is meant to be your only study material, so it is comprehensive and includes 2 to 3 previous year questions with answers in years mentioned. Orientation. CSIR-Net Life Sciences lecture. Orientation is a fundamental behavioral process by which an organism positions itself in relation to external environmental cues. It is the mechanism through which animals determine and maintain proper direction while navigating their habitat or during complex behaviors such as foraging, migration, homing, or escaping predators. Orientation is not merely random movement but a directional response to specific stimuli, known as orientation cues, which may be visual, auditory, olfactory, celestial, or geomagnetic in nature. In ethology, orientation serves as a proximate mechanism that supports larger behavioral phenomena such as navigation and migration and ensures the organism's ability to find food, mates, breeding sites, and suitable habitats. There are different types of orientation strategies exhibited by animals. One of the simplest forms is kinesis, where the movement rate depends on the intensity of the stimulus but not its direction. For example, woodlice show increased movement in dry environments and decreased activity in humid zones, helping them settle in favorable moist areas. Taxis, on the other hand, is a directed movement toward or away from a stimulus source. Phototaxis responds to light, chemotaxis responds to chemicals, and geotaxis responds to gravity. One of the simplest Orientation is a fundamental behavioral process by which an organism positions itself in relation to external environmental cues. It is the mechanism through which animals determine and maintain proper direction while navigating their habitat or during complex behaviors such as foraging, migration, homing, or escaping predators. Orientation is not merely random movement but a directional response to specific stimuli, known as orientation cues, which may be visual, auditory, olfactory, celestial, or geomagnetic in nature. In ethology, orientation serves as a proximate mechanism that supports larger behavioral phenomena such as navigation and migration and ensures the organism's ability to find food, mates, breeding sites, and suitable habitats. There are different types of orientation strategies exhibited by animals. One of the simplest forms is kinesis, where the movement rate depends on the intensity of the stimulus but not its direction. For example, woodlice show increased movement in dry environments and decreased activity in humid zones, helping them settle in favorable moist areas. Taxis, on the other hand, is a directed movement toward or away from a stimulus source. Phototaxis response to light, chemotaxis response to chemicals, and geotaxis response to gravity are common examples. Positive taxis refers to movement toward the stimulus, while negative taxis implies movement away from it. More complex forms of orientation involve compass orientation and true navigation, map and compass orientation. Compass orientation allows the animal to maintain a constant bearing in a particular direction using environmental cues such as the sun's position, stars, or Earth's magnetic field. This method is used during migration or long-distance travel but does not provide information about the animal's current location. For example, starlings and monarch butterflies use a time-compensated sun compass, meaning they adjust their orientation using their internal circadian clocks to account for the sun's movement across the sky. Magnetic compass orientation, observed in sea turtles and birds, allows detection of Earth's magnetic field lines to maintain direction over vast distances. True navigation, also called map and compass orientation, is the ability to determine one's current location in unfamiliar territory and navigate to a specific target without relying on familiar cues. This complex behavior is rare and typically observed in homing pigeons, sea turtles, and migrating birds. It requires the integration of multiple sensory modalities and spatial memory. In pigeons, for instance, olfactory cues, the sun compass, and geomagnetic information are all used in a hierarchical manner depending on environmental reliability. True navigation is also demonstrated by salmon, which return to natal streams after years at sea, relying on olfactory imprinting to locate the precise freshwater system. Celestial orientation involves the use of sun, stars, and polarized light. Nocturnal migrants, such as warblers, use stellar constellations as fixed reference points. They are known to rely on the position of the North Star, Polaris, or the rotational center of the night sky to determine their heading. Experiments using planetarium simulations have confirmed that birds orient using celestial cues even when real stars are not visible. Polarized light, invisible to humans, is detected by insects like bees and used to maintain direction under cloud cover by detecting the pattern of light scattered in the atmosphere. Magnetoreception, the ability to perceive the Earth's magnetic field, has been demonstrated in diverse organisms including birds, sea turtles, newts, and even bacteria. It is thought to involve either magnetite-based mechanisms, magnetic crystals in sensory cells, or radical pair mechanisms that depend on light-sensitive proteins such as cryptochromes. Magnetoreception allows animals to maintain consistent orientation even in complete darkness or unfamiliar landscapes. Sun compass orientation requires animals to have an internal biological clock, which adjusts for the changing position of the sun throughout the day. If the clock is experimentally shifted, e.g., by altering light-dark cycles, the animal's orientation will also shift accordingly, proving the dependence of orientation on internal circadian timing. In a classic experiment, starlings that had their internal clocks shifted by 6 hours oriented themselves wrongly relative to the sun, demonstrating the time-compensated sun compass system. Orientation behavior is also developmentally modulated and experience-dependent. Young migratory birds possess genetically programmed directional preferences, vector orientation, but adults may correct their paths based on learned maps and experience, map orientation. This shift from innate to learned behavior is a critical area of study in behavioral ecology. In CSIR-NET Life Sciences, orientation is a frequently tested topic under behavioral ecology, often integrated with migration, navigation, circadian rhythms, or sensory biology. Questions typically test understanding of types of orientation, experimental evidence for orientation mechanisms, and the role of biological clocks and external cues. CSIR-NET Previous Year Questions. Orientation CSIR-NET Deck 2017, Part C. An experiment exposes starlings to a shifted light-dark cycle, resulting in altered orientation during migration. This experiment demonstrates innate orientation only. B. Influence of magnetic field only. C. Role of biological clock in sun compass orientation. D. Lack of orientation mechanism. Answer. C. Explanation. The shift in light-dark cycle misaligns the biological clock, affecting sun compass orientation. CSIR-NET June 2015, Part B. Which one of the following is not used by animals for orientation during long-distance migration? Earth's magnetic field. B. Polarized light. C. Olfactory gradient. D. Infrasound communication. Answer. D. Explanation. Infrasound may be used for communication but not for orientation during migration. Explanation. Infrasound may be used for communication but not for orientation during migration. CSIR-NET Deck 2020, Part C. In a homing experiment, if pigeons are released at an unfamiliar location under overcast conditions and show poor orientation, which of the following cues is most likely disrupted? A. Polarized light. B. Olfactory cue. C. Celestial compass. D. Tactile cues. Answer. C. Explanation. Overcast skies obscure the sun and stars, disrupting celestial orientation. Let me know if you're ready for the next topic: navigation, biological rhythms, sleep and arousal, or any other core concept from CSIR-NET Life Sciences. I'll keep the same standard for each: dense, exam-oriented, with PYQs and detailed coverage. You said navigation. ChatGPT said. Here is your CSIR-NET JRF Life Sciences High Standard Lecture on Navigation, written in dense paragraph format, maximally detailed, no tables, and entirely tailored for the CSIR-NET exam level, based on previously asked questions and core concepts. This is designed to be your only study material, so the lecture is complete from basic to advanced, and includes 2 to 3 previous year CSIR questions with answers and years mentioned. Navigation, CSIR-NET Life Sciences Lecture. Navigation is a cognitive and physiological process by which animals determine and control their trajectory through space, particularly in unfamiliar environments or over long distances. Unlike simple orientation, which helps an animal move in a particular direction relative to a stimulus, navigation involves the ability to reach a specific goal or location, often through previously untraveled territory. It is a complex, evolved behavior that integrates memory, spatial learning, environmental cues, and internal mechanisms such as biological clocks and magnetoreception. Navigation plays a crucial role in behaviors such as homing, migration, foraging, and territorial return, and is a foundational area in the field of behavioral ecology and neuroethology. Navigation strategies are often classified into 2 broad categories: piloting and true navigation. Piloting refers to the use of landmarks or learned environmental features to reach a goal. This strategy is commonly observed in birds, bees, and mammals. For instance, honeybees use visual landmarks to return to their hives, and studies show that displacement or alteration of these landmarks can significantly disrupt their navigation. Piloting is highly dependent on previous experience, spatial memory, and the stability of the landscape. In contrast, true navigation, also known as map and compass navigation, refers to the ability of animals to determine their position relative to a goal without relying on familiar landmarks. This form of navigation is most strikingly observed in migratory birds, salmon, and sea turtles. These animals are capable of correcting their path if displaced, demonstrating that they possess both a map sense, knowledge of current location, and a compass sense, knowledge of direction. In experiments with homing pigeons, individuals released from unfamiliar sites far from their home were still able to return accurately, proving the presence of a complex navigational system beyond landmark use. Several types of environmental cues are used in animal navigation. Celestial cues, including the position of the sun, stars, and polarization patterns of light, are widely used by birds and insects. Sun compass orientation requires animals to use their biological clock to correct for the sun's movement during the day. In an iconic experiment, if the circadian rhythm of a pigeon is artificially shifted, it orients incorrectly with respect to the sun's position, proving that sun compass orientation is time compensated. Similarly, stellar navigation is used by nocturnal migrants such as warblers, which rely on the fixed patterns of stars near Polaris for directional reference. Another important cue is the Earth's magnetic field, perceived through a process known as magnetoreception. Some species of birds, turtles, amphibians, and even bacteria use magnetic field lines to guide their navigation. Two mechanisms have been proposed: magnetite-based detection involving magnetic particles in the body that align with Earth's field, and light-dependent radical pair mechanisms involving proteins like cryptochromes in the retina. The integration of visual and magnetic inputs suggests a multimodal sensory map of the environment. Olfactory navigation is used by many animals, especially those that return to precise locations such as salmon to natal streams, or pigeons to home lofts. In salmon, olfactory imprinting during early developmental stages allows them to detect specific chemical signatures in water years later, guiding them during spawning migrations. In pigeons, experimental blockage of the olfactory system leads to loss of navigational accuracy, indicating that smell plays a crucial role in forming a cognitive map of the area. Infrasound and acoustic cues are used. In pigeons, experimental blockage of the olfactory system leads to loss of navigational accuracy, indicating that smell plays a crucial role in forming a cognitive map of the area. Infrasound and acoustic cues are used by some animals to detect low-frequency sounds that travel long distances. Elephants and pigeons may use these sounds to gain environmental orientation. Other cues include barometric pressure, polarized light patterns, and even gravity gradients, although their roles are less well understood. In terms of development and evolution, young animals often show genetically encoded directional behaviors, as in young migratory birds, while older individuals fine-tune these innate maps through learning and experience. This interaction of instinct and learning is a central theme in understanding animal navigation. For example, black-capped warblers bred in captivity show genetically inherited migratory directions, but their offspring can eventually adjust and correct routes through experience and social learning. The neural basis of navigation includes structures such as the hippocampus in vertebrates, responsible for spatial memory and cognitive mapping. In birds, hippocampal volume correlates with navigational complexity. In insects like bees, the mushroom bodies serve similar roles. These neural adaptations allow animals to build internal representations, maps, of their environments and make decisions based on past experiences and current cues. Navigation also involves error correction mechanisms. When displaced, an animal compares expected cues with current environmental input. If there is a mismatch, it alters its path until correct alignment is restored. This feedback-based correction is evident in the looping flight patterns of homing pigeons, where they sample cues before committing to a homeward trajectory. This topic is frequently tested in the CSIR net exam, often combined with related areas such as migration, orientation, biological clocks, and learning. Questions may focus on mechanisms of navigation, experimental manipulations, specific cues used, or comparison of navigation types. CSIR net previous year questions. Navigation CSIR net Deck 2017, Part C. When pigeons are released from an unfamiliar location and their olfactory bulbs are blocked, they show impaired homing ability. This result supports the idea that a magnetoreception is not involved. B. Celestial navigation is innate. C. Olfactory cues play a role in navigation. D. Navigation is purely instinctive. Answer C. Explanation. Blocking olfactory input disrupts navigational performance, highlighting the role of smell. CSIR net June 2013, Part C. Which of the following is essential for time-compensated sun compass orientation in birds? A. Magnetic particles. B. Pineal gland activity. C. Biological clock regulation. D. Lunar cycle entrainment. Answer C. Explanation. Biological clocks allow birds to adjust for the sun's position across the day. CSIR net Deck 2022, Part B. Sea turtles use which of the following cues to navigate across oceans to their nesting sites? A. Sun compass and olfactory gradients. B. Magnetic cues and ocean currents. C. Infrasound and auditory landmarks. D. Solar heating gradients and chemical signals. Answer B. Explanation. Sea turtles use geomagnetic fields and ocean currents during long-distance navigation. Let me know when you're ready for the next topic: biological rhythms, sleep and arousal, learning, or anything else from the CSIR net Life Sciences syllabus. Each lecture will follow the same quality and format. You said. Domestication and behavioral changes. ChatGPT said. Here is your CSIR net JRF Life Sciences high-level lecture on domestication and behavioral changes, written in detailed paragraph format, maximum word coverage, no tables, and directly aligned with CSIR net previous year question trends. It includes 2 to 3 CSIR net questions with answers in years mentioned. This lecture is tailored as your only study material, fully covering base to advanced content needed to crack the exam. Domestication and behavioral changes. CSIR net Life Sciences lecture. Domestication refers to the long-term, selective breeding of wild animals and plants to adapt them to human needs. In animals, domestication involves the modification of behavioral, morphological, and physiological traits, making them better suited to living in close association with humans. This evolutionary process, shaped by both natural selection and artificial selection, results in profound behavioral changes compared to wild ancestors. The phenomenon is a critical topic in behavioral biology and evolutionary ecology, with implications for genetics, neurobiology, and ethology. At the behavioral level, domestication typically leads to reduced fear responses, increased sociability, reduced aggression, and enhanced tameness. These changes stem from selective breeding for traits that make animals easier to manage, breed, and coexist with humans. A classic example is the domestication of wolves, Canis lupus, into domestic dogs, Canis lupus familiaris, where selection for tameness inadvertently led to a suite of changes known as the domestication syndrome. These include behavioral traits, e.g., docility, attachment to humans, physical changes, e.g., floppy ears, curled tails, and developmental shifts, e.g., prolonged juvenile behavior and neoteny. Domestication affects neuroendocrine pathways, particularly those regulating stress and fear. The hypothalamic pituitary adrenal HPA axis, which mediates the stress response, is often downregulated in domesticated animals, making them less reactive to environmental stressors and more tolerant of human proximity. This alteration reduces cortisol levels, suppresses fight or flight behaviors, and facilitates bonding with humans. In contrast, wild animals exhibit high alertness, aggression, and wariness due to a more active HPA axis. Behavioral changes under domestication can be either direct targets of selection or indirect consequences of selection on tameness. Domestication refers to the long-term, selective breeding of wild animals and plants to adapt them to human needs. In animals, domestication involves the modification of behavioral, morphological, and physiological traits, making them better suited to living in close association with humans. This evolutionary process, shaped by both natural selection and artificial selection, results in profound behavioral changes compared to wild ancestors. The phenomenon is a critical topic in behavioral biology and evolutionary ecology, with implications for genetics, neurobiology, and ethology. At the behavioral level, domestication typically leads to reduced fear responses, increased sociability, reduced aggression, and enhanced tameness. These changes stem from selective breeding for traits that make animals easier to manage, breed, and coexist with humans. A classic example is the domestication of wolves, Canis lupus, into domestic dogs, Canis lupus familiaris, where selection for tameness inadvertently led to a suite of changes known as the domestication syndrome. These include behavioral traits, e.g., docility, attachment to humans, physical changes, e.g., floppy ears, curled tails, and developmental shifts, e.g., prolonged juvenile behavior and neoteny. Domestication affects neuroendocrine pathways, particularly those regulating stress and fear. The hypothalamic pituitary adrenal HPA axis, which mediates the stress response, is often downregulated in domesticated animals, making them less reactive to environmental stressors and more tolerant of human proximity. This alteration reduces cortisol levels, suppresses fight or flight behaviors, and facilitates bonding with humans. In contrast, wild animals exhibit high alertness, aggression, and wariness due to a more active HPA axis. Behavioral changes under domestication can be either direct targets of selection or indirect consequences of selection on tameness. For example, reduced aggression and increased sociability may have been directly selected, while other traits like changes in coat color or ear shape may arise as pleiotropic effects or via changes in neural crest cell migration, which affects multiple systems during development. This was experimentally demonstrated in the long-term domestication experiment in foxes conducted by Dmitry Belyaev in Russia. By selecting only the tamest individuals for breeding over generations, researchers observed not just reduced aggression but also the emergence of physical traits associated with domesticated animals, such as spotted coats and curled tails. Domesticated animals often show enhanced capacity for social learning and communication with humans. For example, dogs are capable of interpreting human gestures like pointing in gaze direction, skills rarely seen in their wild counterparts like wolves. This suggests that domestication not only alters emotional and stress responses but also enhances cognitive and communicative skills that favor human-animal interactions. On the contrary, domesticated animals may also show reduced problem-solving abilities or vigilance, as they are no longer under constant predation pressure or environmental challenges that drive complex behaviors. Domestication may also influence reproductive behaviors. In many domesticated species, seasonal breeding is lost, and animals can breed year-round due to human-controlled environments and artificial selection. This increases reproductive success and population management. However, these changes can lead to maladaptive behaviors if animals are released into the wild, where domesticated traits may reduce survival. Domestication also affects vocalizations, with many domesticated animals producing more frequent, varied, or higher-pitched calls compared to their wild relatives, possibly to facilitate better human-animal communication. In some species, such as domesticated cattle or pigs, behavioral changes include loss of territory defense, group cohesion, and predator avoidance behavior. These alterations are not always beneficial for the animal's fitness in natural settings but are adaptive in the context of human management and captivity. Importantly, domestication is not synonymous with taming. Taming refers to the behavioral adaptation of a wild individual to humans, often through learning, without genetic change. In contrast, domestication occurs at the population level and involves heritable changes accumulated over generations. Therefore, domestication is both a genetic and behavioral phenomenon, reflecting evolutionary transformation under human control. From an evolutionary perspective, domestication represents an artificial co-evolutionary. Therefore, domestication is both a genetic and behavioral phenomenon, reflecting evolutionary transformation under human control. From an evolutionary perspective, domestication represents an artificial co-evolutionary process, where humans influence the evolution of animals and, in some cases, animals influence human behavior and culture. Dogs, for instance, are believed to have co-evolved with early human societies, affecting hunting, protection, and even social organization. The CSIRnet often explores this topic through questions on behavioral traits altered due to domestication, neuroendocrine control, experimental evidence like Believ's foxes, and differences between taming and domestication. CSIRnet previous year questions, domestication and behavior, CSIRnet June 2015, Part C. In Believ's silver fox domestication experiment, selection for tameness over generations led to increased aggression and territoriality. B. Loss of reproductive behavior. C. Emergence of juvenile traits like floppy ears and altered coat colors. D. Enhanced foraging strategies in natural settings. Answer C. Explanation. Selection for tameness led to neotenous physical and behavioral traits, confirming domestication syndrome. CSIRnet Deck 2018, Part B. Domesticated animals generally exhibit which of the following compared to their wild counterparts? A. Increased HPA axis activity. B. Higher cortisol levels in stressful environments. C. Reduced fear responses and higher sociability. D. Greater predator vigilance. Answer C. Explanation. Domesticated animals show reduced stress reactivity and enhanced social behavior toward humans. CSIRnet June 2021, Part C. Which of the following statements best distinguishes domestication from taming? A. Domestication occurs in individuals. Taming at the species level. B. Taming results from heritable genetic changes. Domestication does not. C. Domestication involves selection across generations. Taming is learning within a lifetime. D. Domestication always increases brain size. Taming reduces it. Answer C. Explanation. Domestication is a population level. Heritable change. Taming is individual level learning. Let me know if you want the next lecture, perhaps on biology. Which of the following statements best distinguishes domestication from taming? A. Domestication occurs in individuals. Taming at the species level. B. Taming results from heritable genetic changes. Domestication does not. C. Domestication involves selection across generations. Taming is learning within a lifetime. D. Domestication always increases brain. A. Domestication occurs in individuals. Taming at the species level. B. Taming results from heritable genetic changes. Domestication does not. C. Domestication involves selection across generations. Taming is learning within a lifetime. D. Domestication always increases brain size. Taming reduces it. Answer C. Explanation. Domestication is a population level. Heritable change. Taming is individual level learning. Let me know if you want the next lecture, perhaps on biological rhythms, reproductive strategies, neuroethology, or any other CSIR topic. Each note will be equally exhaustive and tailored to your prep. Search. Reason. ChatGPT.